the next two slides to the third. This is kind of a slide that's similar to the one that Professor Weber showed. There we go. Thank you very much. And here's the idea. Suppose you have an irreducibly complex molecular machine made up of 6 different parts, labeled A to F. Suppose that they started out as individual precursors, each of which had their own function. Could you, next slide please. Now suppose they got together somehow. Now you could have an irreducibly complex machine, the story goes, but each of the parts had their own function. And I submit that even if all of these things are doing the exact same thing they would do in the irreducibly complex machine, you still can't get from these parts to here, even if they're doing the same thing. And why is that? Well, here's one reason. Proteins are not little colored squares.
Exactly.
They are not little colored figures like Ken Miller showed on his slide. Why does the A go next to the B here, besides being in the alphabet there? Why doesn't it go down here? Why aren't these in a line instead of something like this? What makes a real protein stick to another real protein, and another real protein stick to that? They stick to each other because they have very complex surfaces which are matched to each other. The chemical and physical properties have to be just so, like little magnets that are lined up in order to allow them to bind. Could I have the next slide, please? This is the real problem that would face that scenario. You would have proteins, A, B, C, D, which did not have the right shapes in order to bind to each other. In order to form an irreducibly complex molecular machine, they would have to pre-adapt all of their shapes in order just to bind to each other.
Michael?
Yes.
Again, I would like you to address the issue. What I was talking about were not only hints from the comparative sequences, but the fact that we have the technology now to take those genes and get putative ancestral sequences, mutate them, find out how the proteins might fit together, look at perhaps functional intermediates that are along that pathway that you put the X's on. And what do you tell the scientist who's pursuing that kind of program that will give them a better research program that they should abandon that approach for an ID approach?
I don't think they should abandon it. I just say that they aren't doing a Darwinian approach. They're simply looking for different structures and then ascribing the results to Darwinian processes.
Well, I was